Ready? Okay, so I'll start. Okay. Good morning, afternoon, evening, hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you for joining us, the 34th seminar. Oh my God, already 34th seminar. And I call it DOE JGI Day. I started my 10 weeks of seminar trips yesterday, with the first stop being Stanford University, California, to give my first seminar, this crazy 10-week trip. I'm joining SynBYSS right now from UC Berkeley. And actually our pioneer speaker, Yasuo Yoshikuni, is from DOE JGI, which is actually located in Lawrence Berkeley National Lab, of course, in Berkeley. So I'm talking at 4 a.m. with Yasuo from the technically same place. So, okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Yasuo Yoshikuni. He is the DNA synthesis science program head supporting many researchers in synthetic biology who need massive DNA synthesis capability for synthetic biology research. So we are very grateful for his service to the community. I'm also studied a 500 kilobase DNA synthesis project this year. So again, you know, I personally very grateful for his and his team support. In addition to this amazing community service, he's also performing fantastic research, including the development of chassis independent recombinase assisted genome engineering called CRACE, and actually working for many different microbes. And that's amazing technology you may want to try. So with that, you know, Yasuo, the virtual podium is all yours. And thanks so much for your time and support for the synthetic biology community. Also, thank you for joining us in this early time of the day. And thank you.
Thank you, Taesok. Thank you very much for invitation and nice introduction. Can you hear me okay?
Yes.
Okay, let me share my slide.
Wonderful.
Yeah. Okay, great. Can you see the slide? Can you hear my voice?
Yes.
Okay. Okay. Yeah, again, thank you very much for the invitation and nice introduction. My name is Yasuo Yoshikuni. I'm a head of DNA synthesis science program. It's my pleasure to share some of the stuff that I've been working on at the Joint Genome Institute, JGI. So let me first explain a little bit about the DOE Joint Genome Institute, JGI. This is our building, Integrative Genomics Building, IGB, which is located LBL campus up in the hill of UC Berkeley. Our mission is to provide the global research community with the free access to large-scale DNA sequencing and DNA synthesis technology in order to tackle DOE mission-relevant research project that you're interested in. JGI is established in 1997 in order to tackle a human genome project. After we completed reading the 3 chromosomes of the human, we converted into the user facility since 2004. We are located at the Lawrence Berkeley National Lab, as I mentioned. We have about 300 staffs and $80 million annual budget. And we have 2,000 global primary users and 10,000 data users. So DNA synthesis science program, which is essentially a grant program, excuse me. Our mission is to harness the power of DNA synthesis, strain engineering, and biosystem design for DOE mission-relevant discovery and application. We have a open call, biannual open call. Sorry. CSV functional genomics, and this is biannual. The next deadline is July 31, 2022. We actually don't provide the soft money, but we provide the large-scale DNA synthesis, like a synthetic biology capability support. For example, like we can provide the 500 kilobases of DNA synthesis, construct assembly, fast-track metabolic engineering, and sgRNA library, and so forth. Any audience, any people in the audience are eligible to apply for this program. Grad students, postdocs, early career professors are welcome to apply for this program. And there are a number of users who are grad students and postdocs as well. So regarding the output, we've already completed 250 projects and published over 150 publications, including Nature Science and Nature sister journals. And this is our automated DNA synthesis platforms. So I'd like to introduce some of the collaboration that we did with the users and then recent publications. So this is a project in collaboration with Steve Bleezer's group and the Paul Adams group. We've been exploring diversity of enzyme function in glycosyl-hydrolase 94 family. This GH94 family enzymes are unique because it can actually synthesize the oligosaccharide as well as it can degrade the oligosaccharide into a monosaccharide. The first thing that we helped is creating a genome mining, essentially sequence similarity network, and then did the genome mining. And essentially we selected 100 genes in order to maximally sample through the sequence data space. And then we synthesized, delivered those genes to Steve's, and then Steve overexpressed and characterized each of the enzymes for their substrate specificity. It turned out that he was able to find this unique enzymes right here can take gluconac and gluconac 1-phosphate as a substrate, and then concatenate that into acrylolactane. This is a new polymers. Usually like gluconac forms chitin, but then this enzyme can form 1,3 beta rather than 1,4 beta linkage, so that it makes a new polymer, and which Steve named the acrylolactane. In collaboration with the Paul Adams group, we are also able to get the crystal structure of these enzymes, and then started understanding details about these enzymes and molecular mechanisms. The next story is a collaboration with the Mike Kopke at the Tech and Mike Jowett at the Northwestern University. You may be familiar with the paper that recently published in Nature Biotechnology. In this project, they had the collection of industrial Clostridia called David Jones collections. We essentially sequenced, this is about 300 species collection of the Clostridia, and then we sequenced all of them, and then done the genome mining and DNA synthesis. And then the team did the pathway optimization, strain optimization, process optimization. Eventually, they are able to generate the strain that can produce IPA and acetone in the high level and reproducible in the large scale. With the LCA analysis, they are also able to demonstrate that the process using Clostridia actually can reduce the CO2 emission dramatically compared with the existing technologies. So not only the science that we support through programs, my group also had my own research projects, and those are mainly classified into 4 different categories: plant and utilization, microbiome engineering for biomass generation more sustainably, and biofuel and bioproduct formation, hybrid biomaterial synthesis for the product synthesis. As Taesok mentioned, my group has developed a universal strain engineering toolkit called the CRAGE, which allows us to domesticate 60 species across multiple phyla. And centered around this technology, we also apply these kind of technology to do a metabolic engineering, protein engineering, and some of the paper are published in the high-profile journals. Currently, my group is interested in looking into AI/ML guided predictive biosystem design, and I am looking for a talented postdoc. Yeah, I mean, if you're interested in, please feel free to contact me. Also, the program, DNA synthesis program, if you're interested in applying for a DNA synthesis program, the first step is to contact me. So my contact info is right here. I would highly encourage everyone to contact me to further discuss the opportunities. With that, this is the end of my talk, and happy to take any question if I take any.
Wonderful. Thank you. Thank you. May I join you as postdoc? Am I qualified?
I think you're overqualified.
Okay. Okay.
I also have that feeling. Yes.
I'm cheap, by the way. So anyway, so thank you so much for your support again.
But you're a user, one of the users, right?
Oh yeah. I'm sort of kind of working with you, right?
Yeah.
Yeah. So so I mean, I want to emphasize again, people in this audience, YouTube video, audience, especially young people, please submit your proposal to him to get free 500 kilobase DNA. And that submission deadline again is, I believe, the July 31st this year. And what I'm saying here is what can be wrong with the 500 kilobase DNA. You will be able to get many interesting results from huge amount of DNA and their screening. And of course, potentially a top journal publication. So why not? So just do it right now, today. And
can I also add one comment?
Sure.
So yeah, this program is essentially a grant program. So it becomes part of you for your career development. If it's, it's graduate students or postdoc, this is kind of equivalent to fellowship to your work. And if you're early career professors, we are happy to help for your tenure processes. We can convert the DNA sequencing number to a dollar number. So you can also pitch that you got this grant to your university during the tenure process. Yep. And yeah, we are looking for super users and any users who can leverage our technology to bring their science to the next level. So yeah, be great if people can contact me.
Sure. So actually, you know what? I calculate how much money I'm getting, in terms of DNA. That's more than, I think, $50,000 minimum. And then good thing is no indirect cost for the DNA. So there is an overhead, and then that's kind of equivalent to $100,000. And then people do not order that much DNA if that's not free. And then that's one of the great things we can do. And then again, I mean, what could be wrong? I mean, with that many DNA product, and you will generate a lot of great data after that. So I strongly encourage many people, apply that one. I actually didn't apply earlier, and I regret, but I didn't think about seriously why I didn't have the right project to do for that, DNA. So I'm excited to, and then we already kind of starting, multiple different projects out of this one. And then we are thinking about Nature paper or science paper already, even before we getting the DNA. So I'm excited. Thank you so much.
Yeah, thank you. I think that's really a very good opportunity.
Yeah. Okay, so now the main speaker of today with a longer introduction. Professor Fuqing Wu is an assistant professor, actually brand new assistant professor at the University of Texas Health Science Center at Houston. He obtained his master's degree from Wuhan Institute of Biology and Chinese Academy of Science, and then completed his PhD in Professor Xia Wang's lab at Arizona State University. Of course, one of the best, lab regarding synthetic biology circuit, also, building and some other thing as well. And working on synthetic biology and systems biology. He finished his postdoctoral training in human microbiome and environmental surveillance of infectious disease in Professor Eric Allen's lab at MIT. His current research aims to integrate synthetic biology, molecular virology, and genome sequencing approach to develop early warning systems to monitor the emergence and spread of viral disease in the population and explore the impacts of viral infection and treatment on individual and population health. You know, we don't need to emphasize anymore about how important virus were after 2 years of pandemic. And of course, another important part of, you know, my introduction here is I met him for the first time virtually on my wife's birthday last year during SynBYSS informal chat session. Interestingly, his kid's birthday is the same as my wife's birthday. So my question is, what is the probability of two people meeting each other for the first time on their beloved person's birthday? I'm talking about for the first time virtually, and then we didn't know each other. And I strongly believe people are all connected to each other. And if people consider such connection in mind, there would be no war or hatred we currently seeing, you know, worldwide. And of course, I mean, his talk may answer my question as well. With that, you know, Dr. Wu, please take it away. It's all yours. And thank you so much for your time. And then, you know, I'm excited to hear.
Okay, yeah. Thank you very much, Dr. Wu. Thank you. I'm really appreciated that you gave me a very nice introduction, and especially about this great platform. I think it's a very good opportunity for us, for the young scientists, that we can communicate our results and ideas, and it feels very bonded to the synthetic biology community. I think that's the most that's kind of answers the question that this is the chance. It's probably because we are all connected to the synthetic biology community, and then bring us together. With that, so I would like to start my talk. So today I will talk about my work experience in synthetic biology and environmental surveillance work for this pandemic. And yes, so the first part is synthetic biology. So I did a synthetic biology work in Dr. Xiao Wang's lab at Arizona State University. And one of the very strong strengths of this lab is we can combining synthetic gene circuit with mathematical modeling. And the first thing that I was very attracted to is how we can use mathematical modeling to understand, and the synthetic gene circuits to understand how cells make decisions. So in terms of cells, as like even right now, we're making decisions every moment, every day. Like for example, right now you may be thinking, oh, this talk is interesting, so I want to stay, and this talk is not that sounds fun, so I will probably go. So, and like the cells, like for example, in the cells, during the cells differentiating in the cell development, like a stem cell can traverse in a landscape, and then can differentiate into multiple different cell types, and that we call multi-stability. And this is landscape obviously is related or even determined by the genetics or genes and gene networks, and also the environmental stimulus. And one of the real example is the nanolog and GATA6. And these two are one of the master transcription factors that regulate in cell differentiation to determine cells go to like epiblast cells or like endodem cells. And so what we are interested in, we are trying to use, based on this real example, then we abstract this topology, which is X and Y. X and Y can be two nodes, can be represented one gene or multiple gene, as long as they maintain the similar relationship. So in this topology, you can see X and Y are mutually inhibiting each other. But in the same time, they also have auto regulations, positive feedback, and this we call the mean topology. So how we're gonna construct experimentally to construct this circuit. So we use a two hybrid promoter, P-ARA-LAC, which can be activated by ARA-C gene, and it can be also inhibited by LAC-I gene. And another is P-LUX-TAT. This promoter can be activated by LUX-R, but in the same time can be inhibited by TET-R. To visualize how the output of this circuit, we use GFP and mCherry to kind of report the two promoters' activities. And as you can see, like ARA-C can activate P-ARA-LAC, which is self-activation. And at the same time, this can be inhibited by LAC-I, so Y can inhibit X. And at the same time, like LUX-R can self-activate itself and also inhibited by the X. And also we have 4 different inducers to control this circuit to make sure to let us have a controllability on this circuit and see how it behaves. So the first thing we demonstrated that this circuit, we put this circuit into the cells, and what we found is this circuit can drive the cell have 4 different states, which is low GFP and low mCherry state, which is here, and or either high GFP but a low mCherry state, and low GFP but high mCherry state, and also both high GFP and high mCherry state. So there are 4 different stable steady states. And then we developed an ordinary differential equation model to understand how this, which is bifurcation analysis, which is like we see how this cell states transitions changes with some changing parameters. Here is we combine 2 autoinducers to as well as the parameter, and this is GFP and this mCherry. You can see from this diagram is starting from C1 concentration 1, C1. The black bar means the low GFP, low mCherry state. And if we increase in this increase in this parameter, you can see the cell states are changing. So for example, at a C2 concentration, you will see we're supposed to see 4 different states, which is low GFP, low state, and GFP state, and also the red is the mCherry state, and also the golden state, which is the high GFP, high mCherry state. And continue, if we increase to C3 concentration, we will expect to have 3 states, which is the low state, mCherry state, and the high state. And very high concentration of AR of this inducers, we'll only have 1 state left, which is the high GFP and high mCherry state. So this is the bifurcation analysis. So the next thing, how we're going to verify this modeling result. So we did a hysteresis experiment. So first, we're starting from cells, starting from the initially low GFP, low mCherry state, which is here. And you can see here at a C1 concentration, we have good match with the modeling result. At a C2 concentration, we're expecting 4 different states together, but we only observed 3, which is the low state, mCherry state, and a high state. At a C3 concentration, we will have 3 populations, which is the low, mCherry, and high state. And C4 concentration, we only have the high GFP, high mCherry state. So this result shows that we have at 3 different concentrations, so we can have our experimental results and the modeling results are consistent, but in one of them we are missing 1 GFP state. And for the other part of the verification of the hysteresis, we're using the same cells, but using the cells with different state, which is here, the high GFP, high mCherry state. And we do exactly the same induction experiment. And what you can see from the bifurcation diagram is, if we're starting from here, because the stability of the high state, it crosses the whole 4 different concentrations. Basically, all of them will be the high stable and high GFP, high mCherry state. So in the experiment, we also observed the same thing, which is the cell state becomes very stable, and it's hard to transition to other states. So this experiment shows that our circuit can have the potential to generate 4 different stable states, And we verified our modeling result, but we're still missing the 4 stable state, which. And if we take a careful reading of this bifurcation diagram, you can see if the GFP state, which is the green ball, it has a relative very smaller parameter range, which means this GFP state may be not that stable, or maybe it's very hard to achieve the GFP state in experiments. So the next thing is, based on this observation, we are arguing that maybe we should push the cells to the GFP state first, and then go to the and that way can help the cells to generate false populations. And that is exactly what we observed in experiment. So we try to use the 3 different inducers, triple induction, and you can see from 6.5 hours, the cells are starting to transition to the GFP state, and then you go to 3 populations, and then at 24 hours around, it will have 4 different populations. So these are saying that we can achieve this quarter stability or quarter stable steady states from the triple induction. And we also did some quasi-potential landscape analysis for this circuit. And what we see here is here is the cell state without any induction. You can see we have a very deep value, which is a low GFP, low M chair state. And if we induce the cells with the triple induction, you can see that the landscape will change it to 4. You can see right now, you can see 4 of the values, which means the cells can have 4 different stable state states. And we experimentally, that's what we observed. So there are other ways. The other way is we're sequentially adding AHL ADC, which is to promote the cells to GFP state. And then we add the other inducer to push the cells to other 2 state, which can be also realized into the 4 stable states. And we also verified that in the experimentally. And there's another way is we push the cells to M cherry first, and then transition to the GFP and high GFP, high M cherry state. And what we saw here is you can see this valley, which is the M cherry state. This valley becomes too deep. It's relatively too deep that the cells make cells hard to jump out to other states. So if we doing this way, this induction strategy, so it's very possibly will be adding have GFP M chair state and low state, which is also we experimented observed this. So this result showing that we can change the landscape sequentially, and then use that to guide the cells to differentiate into different populations. And we also verified our result using the microfluidics, and at the single cell level, you can see the cells start transition to the GFP state, and then go to the high GFP, high M chair state, and then some cells will go to the M chair state. So I go to, and then the next thing is based on this mean circuit, when the next question we're asking is whether we can introduce some cell communication into the circuit, which is like XY can mutually inhibit with each other, and then they also have the auto regulations. But in the same time, also XY can kind of have mutual activation through kind of a cell communication. And here's the top circuit design. And so I know it's too complicated, so I'm not introducing the detail of this circuit, but you know what I mean is like X and Y can be also like communicated with 2 different autoinducers, which is very widely used in common sensing components. So with this one, because they can X and Y can also communicate with each other. So we're thinking might be we can use this circuit to generate some spatial pattern. So the first thing we did, we grow the cells in agarose pad and see the cells how it grows at the microscopic level. And here is the time cost result. You can see initially is GFP, and then at about 12 hours, you can see some cells, there are green cells and red cells. And even at some time, you can see some green cells and red cells are kind of lined against each other. Like for example, here, the green cells, red cells, green cells, red cells. It's like so it's very good, well aligned with each other. And but at the end of the experiment, about 21 hours, you can see this the cells form some kind of a colony, even if it's microscopic level. And you can see that the major one is like in the middle is mostly red cells, and the other side is a green cells. So they're forming some kind of a ring pattern. And so we also like verified our experiments using on growth bacteria in the petri dish, and at about 24 hours, we also observed that there's a GFP, which is like a GFP ring, but inside is cherry. So this kind of replicates our results at the microscopic level. This is basically showing that our MIMPAC circuit can kind of direct the cells self-organize into some ring pattern. So the next question is obviously like how we can understand this. We see the interesting phenomenon, but how we do not understand. So if we demultiply this our MIMPAC circuit, you can see it is composed by two symmetric submotif, which is X, Y X can positive feedback for X, and in the same time it can promote Y expression, but Y can inhibit X. So this is the very classic oscillation topology, which is a negative and positive circuit. But as we know that this classic oscillation topology is this oscillation. If we want to generate oscillation based on this, it's very important for us to have a good control for this the strength of the negative feedback. Like for example, if the negative feedback strength is too small, which means X will keep producing, but Y the inhibition from Y is very small, so you will not have oscillation. And if the strength of negative feedback is too big, which means you will have a damped oscillation. So, and that is exactly what we see from this the simulation result. You can see only at some range of the negative feedback strength will have oscillation, but after that the oscillation will disappear. However, if we're using the same parameter set for the MIMPAC circuit, you can see we have no matter the strength of the negative feedback, we have a very robust against the noise and generated the oscillations. So from this, we kind of think maybe our MIMPAC can help to cells to generate a more robust oscillation. And as we are thinking from the cells, like it divides from one cell and then divide and then grow more, and during growing, the colony is also expanding on the agar plate. So that's kind of we have a oscillation circuit. The cell's gene expression level is oscillating, but we also have the cell's growth, also the colony expansion from center to outside. And that's, so then with that we did a PDE model simulation, and we see how this concentration changes with the distance and with time. So starting from the time 0, you can see initially we will have one spike for the C12 concentration, which is we represented the GFP. And but with time, you can see we start to see the peaks. And like, for example, at 42 simulation time, we will have the peak here, but at 62 simulation time, we will have another peak here and here. And this is so that's the time equals to 90. And this kind of showing this the wave, the peak is traveling. And that's very related to the very common phenomenon in cell development, which is the somatogenesis. And that's the way that's where like our development is. We have this clock and a wavefront, which is like, and we also we also grow the cells, and you can see with the time that start to, like, for example, 24 hours in experiment, we start to see 2 rings and then 48 hours, you can see the ring is expanding. And so when we analyze this GFP intensity with the distance, you can see this peak is also like at 16 hours, we have a peak here, but at 48 hours, we have two peaks. One is here and one is here, that representing two different strips, two different rings. And at 72 hours, you can see the range is expanding. So this results are kind of match to our PD model simulations. So what we are thinking is this might be this ring pattern formation is because of the cells oscillating circuit, but also connected with the cell growth. And what we call is the clock and wavefront model. Yep, so I will stop there from the synthetic biology project. So the next, I will introduce the wastewater-based epidemiology project that we did for this pandemic. It's all of a sudden and it's not intentional. So what we did is at that time, we are like 2020, and in February 2020, we are asking what we can do for this pandemic. So we are thinking maybe we saw one publication that the SARS-CoV-2 can be shared by in the stool. That means that that means we have a chance to detect this viral particles in the stool sample and also in the wastewater. So, and that basically illustrates the rationale for why we can do this. Is every time we go to the toilet, infected individuals go to the toilet, and then we start shedding the viral particles into the sewage, and then it will finally go to the pipelines, and then will be in the end it will go to the wastewater treatment plants, and depends on the wastewater treatment, the complexity of this facility we have. Here we can collect the samples at the primary tank, which is where the raw wastewater samples come from. It's not treated, and so we collect samples from here, and then we start to concentrate the viral particles, and start to develop a method to concentrate the viral particles, and then do the detection. So surprisingly, what we found is here we analyzed like 10 different samples from middle March to middle March from the Massachusetts wastewater treatment plant. And what we found here is all of samples are positive of SARS-CoV-2, and the concentrations are around 100 copies per mil of sewage. And as a control is we also analyzed the historical wastewater samples in January, and both of them are negative. That means the signals we saw is real, and we also verified with sequencing. So one of the very practical considerations for this wastewater surveillance is because we know the wastewater is like some place can go to raining, go to rainstorm, go to even like a snowstorm, and this kind of things can impact the concentration of the viral particles that we observed, and that especially like for example in some time that were heavy rain, and that rain can kind of some of them will influence into the wastewater pipes, and can dilute this viral concentrations. So we have to have some calibration method to calibrate this data. So the first thing we did is we calibrated our viral concentrations by the flow rate. And after this calibration, you can see that we can see kind of like an increasing trend for this viral concentrations during the early pandemic phase. But there's another problem for this method is we calibrate the flow rate, but we also need to calibrate that the variations from the experiment, especially about the viral concentration process. And so based on that, we found plant virus, which is called PM, is pepper virus, is very abundantly found in human stool samples, and relatively stable based on different time. And so we used that. We verified this the stability of PMMV in wastewater, and then we calibrated the viral concentrations by PMMV. You can see and the increase in trend is more obvious, is much better after this calibration. And it also matched the increase in trend of the clinical cases. So the next question is we're asking what we can use for this data. So as we know that if we have more people getting infected, that means there will be more virus will shed into the wastewater. That would means we would have observed higher viral concentrations. So which, like in reverse, we can kind of using the wastewater data to infer the infection prevalence. And that is based on this formula. We multiply the wastewater concentration, multiply the total flow, and then divided by the average viral shedding per individual per day. And what we found here is this is the cumulative cases, and this is a wastewater-based estimate. You can see that all wastewater data shows that the viral concentration is significantly higher than the compared to the clinical reported data. That I'm indicating that during the early pandemic of time, our reports for clinical infections is much low, underestimated. So the second thing we can do is we try to analyze the samples like by time. This tempo, so we will have a tempo data, and that data can be very useful, especially the first one is we analyze the samples from January to May 2020. And what we found here is the first day that we found the viral particles in wastewater is March 3, which kind of is one of the application of this method that we can analyze the wastewater samples to date. When the virus is transmitted in this in the community. And the second thing what we can do is we can compare the wastewater data trend and the clinical data trend, and which is shows here. The blue one is the viral titer, viral concentrations, and the yellow one is the daily new cases. You can see here the wastewater data, which is the viral titers, the trend comes earlier than the clinical data, which means all wastewater data can kind of provides an early warning is preceding of the clinical reports. So we also did a simple correlation and found the time lag between how many days that wastewater can preceding this clinical reports, and it turns out like about 4 to 7 days. So it's about a week earlier than the clinical report. And if we have a much detailed analysis of the sample of wastewater, then we could we can use this data to kind of predict like how this viral trend will go in this community. And the other application, what we're asking is, so we have the tempo wastewater data, and we also have the tempo clinical reported data. And so we are trying to use these two data sets to infer the viral shedding dynamics. The viral shedding means like after people get infected, and then it might be like, for example, it might be we start to shedding virus at day 1 and then goes high and then gradually goes down. So this kind of shedding profile is dynamic, and we can use these two data. We can build a convolution model to convolute this wastewater data and clinical data, clinical reports, the number of new cases to which correspond to the total viral load. So we can kind of use these two data sets to infer the viral shedding, the average viral shedding profile in this community. And what we found here is we found a very early peak of viral shedding of the SARS-CoV-2. Like for example, here we about day 3, we found a very peak shedding, which means after we get infected, like about 3 days, we will have the peak shedding of SARS-CoV-2, and then it will gradually goes down. And this is very unique for wastewater data because like in clinical data, we are lacking this data, because when people get infected and have symptoms, and then they will go to the hospital to get tested. But we will never know like what's the first few days before they showed up, like how this viral shedding looks like. But with wastewater data, we can have this opportunity for us to understand this profile. And also we know that our clinical report data is underestimated. So we calculated the different underestimation based on the reported seroprevalence estimate. So either 6-fold underestimation or 24-fold. You can see all of them share the same profile. The only difference is the magnitude is smaller for example, for 24-fold underestimation. But they all share the same conclusion is we have a very early burst shedding of SARS-CoV-2 after infected at around 2 to 3 days. And this is kind of consistent to recent report that people, when using the people challenging experiment, that they found the peak shedding is from about 5 days, 4 to 5 days. So we also analyzed, like at that time, we collecting samples from 40 different US states, about 2,000 samples, and we analyzed the wastewater, SARS-CoV-2 in wastewater. And then one of the analysis we did is we can aggregate this data by time and compare with the clinical data. And you can see that the red one is the viral concentration data, and the green one is the reported daily new cases. You can see the wastewater comes way more earlier, about a week earlier than the clinical reports. And that is another demonstration of we can use wastewater as a kind of early warning for this pandemic trends. And we also correlated this, the wastewater concentrate with the total SARS-CoV-2 viral load in the wastewater compared to the instance in this community. You can see we have a very good correlation. That means if we see more people get infected, we also can see expect to see the high viral load in the wastewater. And then based on this data set, we also answer some questions like including like how many people infected in this population that we can enable us to see the SARS-CoV-2 signal in the wastewater. That basically is the detection rate. So what we found here is we found like when there are 13 people get infected, 13 new cases per 100,000 people community, we will have like about 80% chance to see the signal. And if we plot the density of this, the instance rate, the median detection rate would be 1 new cases per 27,000 people. And that kind of showing that's the median detection rate for wastewater surveillance. And we also analyzed this for this detection rate, it also correlates with the served population size as well as the instance. And overall, if we compare these two, we found there are about 60%, 62% consistency between wastewater surveillance data and the clinical surveillance data. So this result basically showing our wastewater surveillance can be very useful as a complementary metric to help understand the pandemic progress. And we also developed some simple metric, like for example, using the wastewater data, wastewater viral concentrations divided by the clinical cases, which is the WC ratio to indicate the testing capacity. And with some more modeling analysis, we found like before August 15, 2020, which is the first wave of this pandemic, our wastewater can precede the clinical data. But in the second wave of the pandemic, you can see the preceding time becomes much more smaller, which are indicating that we have, at least in Massachusetts, have a very good job to testing people and reporting the result. So we also did some sequencing the wastewater, and I will not, we get the samples, we sequenced about 400 different samples, and we using some method, like for example, to filter out to like kind of improve the quality of our data. And we then found a ton of like SNPs and the mutations and indels, and yeah, here's the result of what we found. So we found about 10,000 mutations even for samples before July 2020, and which is about four months during the early phase of the pandemic. And we use that data to compare the clinical reports. And what we found you can see here is the blue dot, which is so the axis is the SARS-CoV-2 genome, and the y-axis is the prevalence of this mutation found in our data set. And you can see we found a lot of SNPs mutations that shows later than clinics, which means we see a mutation in wastewater, but it's already reported in clinics, which is consistent. And we also found a lot of mutations that we found in wastewater and much earlier than the clinical reports. And also there are about 50% of mutations that is not reported in the clinics. And then if we do this, summarize this data, which is the time difference between wastewater finding and clinical reports, you can see most of them are wastewater mutations found much earlier than the clinical reports, even like a year earlier. Like that basically means a mutation found in wastewater in 2020, June 2020, is not reported until 2021, in August 2021, that is reported. And that is kind of showing that wastewater sequencing can also be very helpful to kind of see this, to track this variance. And we also compared that, like assign these mutations to different variants. Like for example, in the samples before the 2020 June, before the June 2020, we've also found the alpha mutation, alpha variant, and a beta variant, and also gamma variant, delta variant. So right now we're talking about the endemic. We also wrote a paper that is to discuss what wastewater surveillance can be used for during this during the late pandemic phase. And that is like, for example, we can monitor the wastewaters, like go to the upstream, which means go to each like building level or school level to monitor this virus. And we can also build some modeling to maybe somehow we can combine the wastewater surveillance and the clinical surveillance to have like to achieve like a more affordable, more cost-effective way to monitor this SARS-CoV-2 infection in the community. So what I talk about two topics about synthetic biology and epidemiology, but what I'm very appreciative, I think like the idea is like maybe we can connect. So right now synthetic biology is everywhere, but in terms of its application in epidemiology, I think it still needs more brains to get in, and especially like besides the technical part, I think the ideas of synthetic biology, especially about the modularity to disciplinary and also the concept to build to understand, I think that can be very helpful for this epidemiology and for the epidemiology field. And for right now, like this field is kind of more about all about the population health, and I think if we can kind of put the strengths of synthetic biology in epidemiology, we can kind of have a better plan that we can help us to better prepare the pandemic, and especially in terms of the early warning and the fast response and like a testing capacity. I think synthetic biology can be very helpful for that. So they are like a little bit about my new lab, which is about, well, focus on the like the wastewater and how we can better improve this techniques, this technology that can be making it more useful for population surveillance, for population health. And also we are doing some like for pandemic preparedness, we need to know how many virus or how many pathogens in the population. So we are trying to also develop method to improve the viral detection and also viral metagenomics to help us to know this viral diversity. And that basically shows the answer of my talk. And we also have one postdoc position that is opening. And if you have experience in genomic data and statistics modeling, and feel free to contact me. Yeah, with that, I would like to thank to my advisor, PhD advisor, Xiao Wang, and which his lab is very awesome. If you are interested in mathematical modeling, nonlinear dynamics, and the synthetic gene circuit, I think that's a very strong place to go. And also Eric Elm in the MIT, and we have a very wonderful team for this wastewater project, and they're very productive and reliable, and it can make this work like it goes very fast. And especially our collaborators at Smart in Singapore and NTU, and I'm still like keep collaborating with them, and they're very reliable, I think, collaborators. With that, I'm happy to take questions. Thank you very much.
Amazing, amazing. I love your talk, especially the both topic is the one of my best kind of interest, and also I'm passionate about this field. So thank you so much for your wonderful talk, and also I love both of your advisors. I mean, they are leading scientists in both synthetic biology and systems biology. So you train very well in both area. So actually, I love it. Let me check whether we have the question. I'll ask question in the case. So this is fascinating. I mean, you use the model and then predict those type of thing. Still, like what I want to ask is, I mean, during pandemic, we collected a lot of data. So probably, prediction could be a little bit kind of assisted because of the huge data set. But, there are many other, problem. So my question is basically using, using model, we may predict many natural phenomena, including climate crisis and tipping point, as well as virus spread.
Yeah.
However, what would be the most challenging aspect when we want such prediction as accurate as possible? Can you kind of give some comment or insight on that point?
Yeah, that's a very wonderful question. So I think one of the challenges for now is how we can like more accurate to convert the wastewater data to the prevalence estimate. Mm-hmm. Like, because right now we have this relative like equations to convert the wastewater viral concentrations to the how many people infected in this community. But that data is I think that formula is way too simple, and we certainly need more sophisticated ways for this conversion, for this to make it have a better understanding of the prevalence. Like right now, what we needed to consider is the virus are degrading in the wastewater, and that degradation depends on time, depends on the travel and distance, and so we need to have we need to consider those factors which will influence our viral concentrations. Mm-hmm. And the other part is like in the experimental side. So right now our viral concentration mass can work. Of course it can work, otherwise we will not detect the viral signal. Mm-hmm. But the concentration efficiency, the recovery rate is super low, mm-hmm. Like probably 1%. I was in 1%, like even people reported like 30% or 50%, but I do think that value is relative, it's not absolute value. So I think we need that there are some more techniques that need to improve on the experimental side with a more accurate wastewater data, and I think, and then we need to think about the flow rate. The flow rate is because it's a population. Like some places may rain, some places not rain, some places like people have a ton of water leading, but some leaking, but some places are fine. So we need to consider this difference, and also different area. We have different different places. Like for example, in Boston, in the wintertime, people stay at home. In the summertime, we go outside. But in Houston, in the summertime, people stay at home because it's too hot. So yes, it's very different. So I think to actually like using the wastewater more reliably, I think we certainly have more stuff to improve.
I see. Interesting. So another core question I also need to ask was your approach. Whether, and I was wondering whether your approach would be generalizable, not only for wastewater, predicting those kind of virus spread, but also thinking about all the data set we currently having, in terms of temperature fluctuation, climate change. Can we kind of predict climate emergency, tipping point, and so on, using mathematical model and data? that if that is possible, I believe that your model or your approach will be very powerful.
Yeah, that's a very good point.
Yeah.
So I think like based on the viral trend, like you can see the changing point, like we see it's shooting high, then we can see it. And then the next thing would be how we're going to quantify that, how we're going to use the smart epidemiological modeling to compare that. So I think that's another area that can be certainly improved. So in my acknowledgement, is Dr. Huang in Arizona State University, and right now we are trying to develop some modeling to compare like before some policy interventions and after. So how the changes.
I see
the reproduction number.
I see. Yeah, so that might be probably that might have the huge impact on any discipline of the science and engineering. So I'm looking forward to seeing more data and more model from you, and I'm so excited. So right now, I already see we have the 6 minute past the basically California time 9 o'clock and 9:06 right now. So we probably need to close. So thank all for joining and staying today, despite currently EBRC meeting happening in UC Berkeley. I need to go there. After the seminar. We'll meet again next week on May 26th, Thursday, the same time, the same Zoom link. But I will be joining from London, UK, after my third seminar or talk during this crazy trip at University College London. And interestingly, we'll have Professor Jeff Baldwin from Imperial College London. And basically, I'm visiting University College London, but I believe they are rivalry, university. And then I will have Professor Ahmed Bedran, who joined Scripps Research Institute in California very recently. So that means another brand new professor. As usual, the follow-up informal chat will occur without recording, and please stay here if you are interested in chatting with us. So thanks. I'm stop recording. Let me stop recording this guy.